Welcome back. This class, we are going to study the types of vaccines. There are three major types of vaccines. They are live attenuated vaccines, non-live vaccines, and nucleic acid vaccines. First, let's introduce live attenuated vaccines. Live attenuated vaccines contain pathogens that have been weakened, altered, or selected to be less violent than their wild-type counterparts. In their altered form, they cannot cause the actual disease or only mimic the disease in a very mild way. Live attenuated vaccines act by causing a very limited type of infection, as the attenuated pathogen possesses the same antigens as the original pathogen. Healthy individuals develop immune response comparable to those induced by the natural infection. So, which vaccines are live attenuated vaccine? For example, measles, mumps, rubella, varicella, rotavirus, herpes zoster, influenza, oral poliovirus, OPV, yellow fever, etc. So, what is the characteristic of live attenuated vaccines? So, the benefit aspects include mimic natural infection and immune response, elicit both antibodies and cell-mediated immunity. It will have lifelong immunity possible after 1 or 2 doses. So, what is the limitations of live attenuated vaccine? The limitations are they have possibility to reverse to natural form, etc. poliovirus. They are less stable over time and heat labile. They are contraindicated for immunocompromised individuals and pregnant women. The second type is non-live vaccines. Non-live vaccines do not contain any living or infectious particles, so they cannot cause disease and cannot reactivate. They generally have a good safety profile even in immunocompromised individuals. However, a drawback of this vaccine is that immunogenicity and duration of protection tend to be less than for live vaccines, because they may require several doses or adjuvants to improve immunogenicity. So the non-live vaccines can contain inactivated vaccine and subunit vaccine. First is the inactivated vaccines. Vaccines based on inactivated pathogens are produced by inactivating preparations of whole pathogens by heat, radiations, or chemicals such as formalin or formaldehyde. Robust technique for production limited immunogenicity adjuvants may be required. So multiple primary and booster doses required to obtain long-term protection, and it has high stability. And inactivated vaccines is no replication of inactivated pathogens, so the pathogen will not replicate. Note contraindicated in immunocompromised individuals and pregnant women, so it is relatively safe. So inactivated vaccines include whole cell hepatitis, rabies, tick-borne encephalitis, Japanese encephalitis, cholera. They are both belong to inactivated vaccines. The second non-live vaccine is subunit vaccines. Subunit vaccine contains selected fragments of the pathogen as antigens instead of the whole pathogen. These fragments can be proteins, polysaccharides, or parts of a virus that may form virus-like particles. We call it VLPs. Subunit vaccines generally cause less advanced reaction than live or inactivate whole organism vaccines, but they may be less immunogenic because they contain fewer antigens and the proliferation process often eliminates components that trigger innate immunity. So the types of subunit vaccine include protein vaccines, toxoid vaccines, VLP vaccines, and polysaccharide vaccines. First is protein vaccines. The antigenic proteins can be purified from preparation of the whole pathogen, as for the acellular pertussis vaccine, or can be produced by recombinant genetic engineering. In the latter case, a gene encoding the antigenic protein is inserted into an expression system able to produce large quantities of the antigen in cell cultures, such as the hepatitis B vaccine, in which the gene of the hepatitis B surface antigen has been inserted into appropriate vectors for production in yeast or mammalian cells. The second subunit vaccine is toxoid vaccines. Some bacteria, such as Clostridium tetani, Clostridium difficile, or Corneobacterium differentium, cause disease by releasing pathogenic toxins. Vaccines against this disease are produced by detoxifying the toxin using heat chemicals. The inactivated toxins called toxoid are no longer pathogenic, but retain their ability to induce toxin neutralizing antibodies. Classical examples of toxoid vaccine are those against diphtheria and tetanus, which have been used since their discovery in the 1920s. The third subunit vaccine is VLP vaccine. VLP vaccines are based on the observation and that expression of a certain viral protein leads to spontaneous assembly of particles, structurally similar to the original virus. VLPs are not infectious because they lack the viral genome. However, the native conformation of the antigenic proteins is well preserved, which improves their immunogenicity compared to the free proteins. The best known examples of VLPs are the human papillomavirus (HPV) vaccines that protect against cervical cancer caused by this oncogenic virus. Currently, licensed HPV vaccines contain antigenic proteins from HPV types responsible for most cervical cancers. The fourth is polysaccharide vaccines. Some bacteria, as Staphylococcus pneumonia, Haemophilus influenza type B, and N. meningitis are three encapsulated bacteria that cause severe invasive disease. They possess polysaccharide capsules that facilitate bacterial survival when carried in the nasopharynx and in the blood during disease pathogenesis. The first generation vaccines against this pathogen were based on capsule polysaccharides purified from whole pathogens such as pneumoconiosis polysaccharide vaccine that was launched in 1983. Okay, this class we know what is live attenuant vaccine and non-live vaccine. Next class, we are going to study nucleic acid vaccine. Okay, see you soon. Bye.